Are we going to be moving that way for quite a while, Chris?
That general direction.
For like hundreds of meters?
Yeah.
Okay.
Where there's going to be a couple mild turns in there, but...
Yeah, that's fine. Okay, so you can, in general, let me get away from you a little and I'll stretch that tether out. So if I get too close to you, I'm only 1 box away. The tether becomes, you know, it can do the 1980s phone cord thing and get tangled up. But in general, we kind of keep Atalanta on the same course over ground that the ship is. So if you want, you can do a couple clicks on +10 and bring Atalanta's heading around to the north and look at Hercules. That should put me back in the box there on the beautiful. And then now that I can kind of, that also gives me some visibility on what's off to my right. I'm like light years up in the water column here. Get back down a bit. If you want, you can do a couple more clicks and bring your heading to 020. So what that does, now I know Atlanta's looking the same way that we're moving. So it does a couple things. It gives me some visibility around and in front of Hercules before we get there. And it also, so I try and stay literally in that box. So if I'm wandering off to the west to smell the flowers, I can see that, you know. So if you change your heading, just let me know. Sometimes you'll change your heading crazy to try and keep Hercules in view. But generally when the boat's moving, we're generally not changing the heading too much. And I tend to get lost all the time. That's why I have a navigator here, usually keeping me in line. So I'm like wandering off to the east there for no particular reason.
You're not going to get too far.
So we want to try and keep your heading 020. And don't chase me around while we're moving.
Ali, front row, could I ask
put another
could do
move
some
in real quick,
introductions?
Danielle.
Roger, another.
Sorry, 1 second.
All right.
Bridge nav, could we do 30 meters 020? Okay, we're good up here.
Hey, who's got control of the cinema camera back there?
We're going to try and get that to be a Harold task.
Harold?
Yep.
You see that image from the cinema camera there?
Yep. Are we going to try and take a picture?
Does it look in focus to you? You
So
can take a fuzzy picture.
So point of question, we were instructed that anything other than taking a picture had to be run by you first.
So there is no one size fits all, and I try not to generalize and make rules in here. But in general, if we communicate, like I just did, not very politely, but you know, you can yell at me too. You are free to play with the focus there and take pictures.
Copy that.
What if Brian's been in here before, he knows. So like with video, we communicate. You know, we ask them to zoom, but Amber's a professional over there, so she's all the time on the focus and the brightness. That's kind of her job. You can play with them as well. The only time it'll really get me is if we're like coming in for a landing and I'm trying to position that. You'll get better still camera photos if the vehicle's not moving. So if I come in, I'll kind of frame it up and try and not be moving the vehicle around. Or I'll just put a toe out and, you know, like put my hand on the handrail to stabilize myself as I'm going up the stairs. and you'll get a much better, crisper picture.
Sounds good. Thanks, Dan.
But they're free, so you can. Okay, Jaden, let's come up a couple meters. So you see that loop right there behind me? I'm lagging here, and more flying, less talking. And then I like to multitask you, and so you can bring your head to the left a little, make it 2020.
Are you ready for a request, Dan?
Sure.
Can we take a look at the little pink thing here, please?
Little pink thing around it. Cup coral. This happens to be my favorite coral.
All right, so as you zoom in here, this is a cup coral. It's one of the deep sea sclerotinians. A lot of the sclerotinians, the actual like hard corals you see in mostly in shallow coral reefs, don't extend very far in the deep sea because of the chemistry. And these little guys, the cup corals that come from a couple different groups, have a better time of being able to calcify or make their aragonite skeletons.
Push in a bit there.
They can be quite difficult to tell apart. So we'll get a quick zoom here, and then I'm hearing the murmurs around from all my fishy colleagues that there might have been a fish over to the left. So once we get a decent little zoom on this, we'll go fish chasing.
So look, if I let go of the controls, Amber gets a really good zoom.
Not so great on the focus part though, we drifted.
I can see, and as soon as I touch it, it's all.
So that was probably good for science. I think there was an eel off to the left we'd like to take a look at,
Okay,
if it's still there. Eel and a shrimp.
Ooh.
That little guy. Sorry, that little critter. Why don't I just say that eel? They do that thing with their mouth, it seems, when the ROV gets close. They get aggravated.
They do it in the Atlantic too. It seems to be some kind of annoyance display. Go for it, Harold. Jump in. What are we seeing?
Yeah, so this is looks like a synaphobranchid eel of some kind. They are scavengers. Jeff and I were actually here in Palau back in March with a National Geographic team where we were studying scavenging animals, scavenging fish, and scavenging invertebrates just like this. And this is a fish that we saw quite commonly throughout both the main island of Palau as well as the southwest islands. And they were quite difficult to distinguish from these baited camera videos because one of the distinctive features is where their dorsal fin ends relative to their anal fin, I believe. And we just got a pretty good view of it there, so hopefully we'll be able to review that footage and get an identification.
I wanted to chase them, but this beautiful coral's on my way to back off.
We're also seeing a lot of different groups of corals here so far on the dive. We have Chrysogorgiidae, these sort of bushy corals that include the spiral iridogorgia here up in the top right, as well as some of these smaller bottle brush type corals. We have bamboo corals, both branched and unbranched. And we've seen a cup coral, the scleractinians. What else have we seen? We've seen a few others,
Several
including
different types of black coral so far.
black corals as well. There's a sea star in the top right. Appears to be different than what we saw earlier. And many sponges. Okay, here comes the
Do we
eel
want to
again.
keep moving, or are we hanging out here for a while?
Keep moving.
I'm pretty
Keep
close,
moving?
so let me stretch it out, Chris.
Okay.
I was chasing the eel.
This is a beautiful spot where you can see all the iridogorgias here, and this larger, probably branch bamboo, could be a primnoid. I think it's a bamboo though.
Come up
here.
5 meters for me, please, Jaden.
Yeah, this coral looks like a bamboo. If you look closely at the base, you can see a node there. I'm trying to look if it's internodal or nodal branching.
Can you explain people what that means?
Yes, sorry, using too many science terms already.
There you
So
are.
bamboo corals, you can distinguish between them, different types of them by looking at where the branches form along the axis of each branch, and they'll have, basically looks like a stalk of bamboo, and the little black proteinaceous part in between two branch segments is called a node. And on some bamboo corals, the branches arise from the node, and on others, the branches arise in between the nodes. And this is a diagnostic feature for bamboo corals.
Can we zoom in on its little friend right here, the squat lobster?
Here.
So another one of my favorite things about this vessel is we have a telestrator like you would use on like Monday Night Football to diagram football plays, where the back row can
Push in a bit there,
point
Robert.
directly on a video and have a little icon pop up that the ROV pilots can hang. And that's I give kudos to Ed McNichol, who helped design this current configuration of the
That's
control room,
good
for
there,
putting that in there.
thanks.
It's something I've wanted for a long time in an ROV control room. And right now this is the only ship that I've sailed with that has it. And it makes life so much nicer.
Yeah, it does.
so
Way
here-
better than a stick.
Here we're looking at a chrysostylid squat lobster living here in this coral. They're a common coral associate. Some species of Chrysogorgiidae almost always have them. They are kind of hit or miss whether they show up in the bamboo corals or not. Right, I think we're probably good enough with this one if we want to make some tracks.
Roger.
Going wide?
Yes, please. Come back around to the north here a bit before we go. Yeah, you can bring your heading to the north, thanks. You just bring your heading to the north. I'm behind you, so we won't spin around. I'm on the wrong side of you.
So I have a question here from a viewer. This was a couple minutes ago, but what is the largest animal spotted so far?
So far this expedition, we
Yes.
are a grand total of maybe 2.5 hours into this expedition. So probably one of those tall iridogorgias, or maybe one of the that pretty big colony of bamboo we saw right there is probably the largest thing we've seen by size. But corals don't weigh much, so that Snaphobranchideo might have been the largest we've seen by mass.
So I would like to share another Palauan word that we can use while we're on this dive. If you see something that's kind of like, wow, you say olek. So if you ever hear me say olek, that means I'm saying wow.
Ole?
Olek with a
Olek.
K at the end.
Olek.
Yeah,
Okay.
so you guys can use that if we see anything amazing
Try
or
and add
crazy.
that to my lexicon.
Thank you so much. I think what we just saw there was a different species of iridogorgia, a magnispiralis. You can tell because it has a less of a tight whorl in its main axis.
is we're seeing a pretty good density and abundance of different corals here. A lot of them are chrysogorgids, but a lot of different species and several different genuses of chrysogorgid. And
close that
looks like
window, will
another good-sized big bamboo coming up here.
That's probably part of the reason we're
So
getting
can you find
liveliness
these
up there.
more in shallow waters as well, or are these just deep sea?
So most of these are deep sea specific. Generally, a lot of people call them deep sea coral, but a lot of times they're also referred to as cold water coral because their distribution a lot of times is temperature limited, not just depth limited. So like in higher latitudes, there are places where you can see stuff that we generally would consider a deep sea coral in much shallower depths because the water's still cold. But yeah, I mean, the depth is the primary zonation force in the ocean. So most of these are somewhat depth constrained.
Yeah, so the depths here are about 2.82 degrees Celsius, and in Fahrenheit for Palawan viewers, that's 37 degrees Fahrenheit. So pretty cold down here, down there.
See anybody home, Harold?
I see one squat lobster, looking to see if there's anything else. There's a crinoid as well in the center.
What is a crinoid?
Yeah, thank you. A crinoid is a type of echinoderm, so it's related to sea stars, sea cucumbers, and sea urchins. And it's something that is sort of more commonly associated with the fossil record. Crinoids are very common throughout Earth's history, but we see them pretty often in the deep sea, and they have their common names. There's multiple kinds, both stalked and unstalked. Sea lilies and feather stars are their common names. What we just saw was a feather star, the ones without a stalk. And here we see another type of chrysogorgid coral, Metallogorgia melanotrichus. And these are notable because they are only ever observed with a single symbiotic brittle star living inside their branches. They have never been observed without this brittle star, even from a very, very early stage of their lives. So sometimes you'll see just the tiniest Metallogorgia coral, and lo and behold, its resident brittle star is still there.
Ready to move, Dan?
Nope.
Say again?
Nope.
Okay.
Sorry, I'm still kind of getting my feet under me here. Comfort level.
Yep, no problem.
We have another question from a viewer for the scientist. How old do you think the big bamboo corals would be?
Hundreds, if not thousands of years old. So some of these, the way their skeletons are constructed does not make it quite hard to date them, radiometrically date them, because they replace some of the proteinaceous parts of their skeleton over and over. But some of the dating work that has been done on black corals indicate that they can be, several thousand years old, and so we expect a lot of these octocorals to be in the many hundreds of years, if not over 1,000 years old. But we don't really have great data on exactly how old that is. But even in thinking about these corals being 600, 700, 800 years old, and how much human history will have occurred in just one lifespan of one coral is pretty amazing. It's also an important thing to think about how old and slow growing deep sea life is when you think about extractive activities in the deep sea. If you knock over one of these corals, it takes 800 years to regrow an 800-year-old coral. And so one moment of human activity can take literally centuries or even millennia to regrow. So this kind of fuzzy thing over here on the left is an old sponge attachment point. The sponge has died and moved on.
Chris, and get it on non-ludicrous speed, like 1.8.
And we've got a couple other different type of glass sponge we haven't seen here yet on this side of this rock.
Yeah, that looks like another euplectellid, possibly
Oh,
Walteria
I forgot
species
about that,
is my guess. It's another one of these glass sponges that sort of forms a cage in which you
Yeah.
can have shrimp and other associates living
I
inside.
forgot about that, bud. Thank you. That's what I wanted. What's this one is? Yeah, they should be on 1.8 all the time. I'm not sure what's going on with that one, why it's not dialed in. Okay. Thank you for reminding me of the, it's like, why? The button gets bumped once in a while. Okay, I'm good for a move now.
Bridge nav, 20 meters, 020. Every time I call the bridge, somebody else is up there.
We need to play with the tri-tech at some point, or it's not very useful at the moment.
I don't. So as we move along here, some more retagorgia. I'd say they seem to be the dominant of the large corals here. Certainly members of the family Chrysogorgiidae make up the majority of the individuals that we've seen here. We've got a couple other primnoids and bamboos floating around as well. Dan, can we take a look at this one?
Sure.
Not sure we've seen that one yet. All right, here comes one of those corals. It's kind of hard to tell apart.
Go ahead, Amber, you can push in there a bit. I gotta find my tilt thingy. One thing you can see, these two buttons here.
All right, Harold, what's your boat?
Ooh, okay, so
Ole.
what we have here is some kind of, could be corallium or hemicorallium, or maybe a paragorgia with, it's got the yellow polyps you see on here are actually not part of the coral itself. Those are zoanthids, which are a parasite, and they will colonize the branching skeleton of their host coral and either slowly replace it or just grow on dead parts. And we also have some ophiuroids in there, but yeah, I'm going to say this is some kind of paragorgia.
Yeah.
There
Are we in a position where you can collect, Dan?
Yeah,
Bridge nav, hold position. No,
that we
we're
are.
already out of the move.
How would you like to collect it, Brian? It's,
Yeah, snip in a bio box, like a decent sized branch with both pink coral and gold zoanthid, please.
Roger. Let me get it. Did you get a picture of it before I sample it?
If you can give us a zoom again, please.
Yeah, go ahead, Amber. You're free to throw your boots there. Not sure why the
And
manipulators.
can we have lasers on for a moment?
Yeah, you can do that if you go to the lights page here. So hotel, lights, and then scale lasers.
Are there different kinds of zoanthids, or are they kind of all the same kind?
That's a great question. There are many different kinds of zoanthids. I'm blanking on what level of taxonomic classification that is, but I believe it is in order. They're more closely related to the scleractinian corals, so your hard shallow water corals, than they are to
We're
the
good with the image, Dan. You
octocorals.
can go for setting up.
What's that?
I said we're good with the image if you want to proceed with collecting.
I'm just thinking here. One of the small boxes in the starboard box?
Yeah,
maybe?
totally. So these kind of pinkish corals are sometimes really hard to tell apart. This is, I'm voting for paragorgid, but it could also be a swiftia. It's moving in the current, so I suspect it's not a corallid of some type because they have a harder skeleton. But we'll know very shortly for sure it's not a corallid here when we just take a piece off.
So I'm going to walk Jaden through this.
It's 100% a paragorgia.
Megan's
Thanks,
hedging there a
Megan.
bit.
Do you want to tell us how you came to that
Stand by,
conclusion?
kids. I'm gonna walk Jaden through
So
what
the
we're
main
doing
character to tell the difference
stand by.
between hemicorallium and paragorgia are looking at the ends of the branches. On the hemicoralliums, there are paired polyps. On paragorgia, there'll be multiple polyps on the ends of branches. And my also fun little nifty trick is if you look at the base of the coral, it tends to look a little bit yellow. That's usually indicative of a paragorgia, whereas the corallium that has a calcareous skeleton will be more of a stark white in color.
Zoom back in, Amber, so we can
Okay,
follow along
paragorgia
with
is
what
yellow
Megan
base.
was just
Got
saying,
it.
because
Thank you.
you're going to have to say that again, Megan, because I wasn't looking while you were talking.
So the little trick for telling the difference between paragorgia and hemicorallium is if you look at the base of the coral, it looks a little bit yellow or brownish, whereas the hemicoralliums will look more white or pink.
But what about swiftia?
Swiftia tends to be, the branching pattern tends to be more 90 degrees, tends to be a little bit more flexible. The polyps happen to, most often happen to be slightly different in color from the main axis. And and the swiftia doesn't have the clusters of polyps on the ends of the branches.
Great, thank you.
Thanks, Megan. Okay, you can stay zoomed in if you want there, Amber. I got 10 other cameras looking at it. I'm just going to walk Jaden through our typical sample scenario. Here, there's a few buttons that they are going to have to press to make this quicker. Right now, we have plenty of time because the boat's not moving, so Atlanta's not trying to run us over. So we'll just take our time and I'll have you do it. So the first thing you're going to want to do is go to Hotel Cameras. And I'm going to ask you to switch these all the time. So you remember our little, 2 cameras into 1 video channel thing? So when we put a sample in the starboard bio box, right now that camera's looking at the port rail, which is the Niskins. That's the kind of blown out, gray things there that are all open. If you turn that one off and turn on starboard bio box cam, now we have 2 images in the starboard bio box. So that one's looking down at it and the other one's looking along it. So that gives me a 90-degree out so I can hopefully get the sample in the box. It's really hard to do with 1 camera. It's like closing your eye and trying to touch your fingers together. Close 1 eye and try and touch your 2 fingers together. I can never do it. Then the other thing we are going to go back to that hydraulic page you were on. So one of the things the most common thing I do here is tilt the camera up and down. So if I need to look side to side, if I can, I'll usually just change the heading on the ROV because it's easier. And I know the camera's always looking forward. But I do tilt up and down a lot because we can't pitch the ROV. So that's what we did when we came in and landed here. I just tilted the camera down. So sometimes, depending on the sample, they may be floaty samples. So I'll kind of like stuff it down in the box and then open and try and get slowly away with the manipulator, but not too fast so I don't suck it out with the water. And then you'll have to like close the box really quick. And sometimes you'll actually close it and capture the sample in the box lid, but at least it's not floating away, and then we can play with it and try and stuff it back in. So the scientists usually know whether they float or sink, and you'll often hear us ask, is it a floater or a sinker? If we have floaties in there, then it makes it really challenging in the future when every time we open the box, the sample we spent some time taking might try and escape. And then in the box, the smaller samples like this we'll put in the back of the box. So if you figure out where the button is there for the starboard box and press that now, you'll see that box slide open. I'm sure we've done this on the deck already a bunch of times. So you can slide it all, you can keep going. There should be some partitions in the back. So those are cleverly labeled some way that I can never comprehend, but I'll usually refer to them as, the forward one or the aft one, inside one, outside one. And then once we're done, you obviously close the box and then change the camera back. So I use that camera to make sure I'm not getting any, too close to big giant corals on the side of the vehicle.
All right, we have a question from a viewer. How do you decide what to sample and what you don't want to get?
So some of it depends on the individual objectives of the dive, and some things are particularly hard to identify, and so we like to get a sample of those. But generally, the way I think of sampling on a project like this is we're looking for voucher specimens. We're looking for potential new records in the area. We're looking for potential range extensions in both depths and geography, and we're looking for, to be able to look at some of these things up close. And so we'll collect one to be able to look under a microscope, potentially do genetics on it, and that helps us as a ground truth or a voucher for all the other things we'll see across the dive and the expedition.
Can you push in a bit there, Amber?
So this, to me,
yeah, keep going.
To figure out what type of paragorgia it is, and then it's kind of a bonus sample here of the zoanthids are chronically understudied in terms of taxonomy, so being able to get both of the paragorgia and a sample of the zoanthids here is kind of a 2-for-1 sample opportunity.
Can you highlight where you'd like to do the snip?
I'm pretty open from your angle. I'm assuming just cutting it across there or something like that would be fine.
Right.
Whatever is convenient for you operationally, that we both get the yellow zoanthid and the pink coral.
Right. I just gotta check the see, grip force. How about we'll try 3. How about grip force 3? How do you feel about that?
So the process is we'll collect it here. We've got some really good imagery on it, and then we'll bring it up to the surface and take more images of it as soon as it gets onto the surface, and then they'll be preserved. And the specimens will be sent to the Museum for Comparative Zoology at Harvard, where they will be kept as part of the museum's collection, and any scientist in the world
there,
can, yep, that looks good, right,
swing and a miss.
can request access to the samples to be able to examine them in their lab and do further work with them. So we're developing a community resource here. And it broke off the bottom. That's too bad. All right, we'll take the whole thing.
Sorry, I hit the console here and didn't get deep enough in my jaws. I did that once with a 1,000-year-old black coral on the side of a cliff in a sanctuary in the Discovery Zone in Canada. And the control room erupted in pandemonium. So basically pulled a Christmas tree off the wall. We wound up, after the commotion died down, they decided, yeah, you can't put it back. And we stuffed the whole thing in the bio box. And, as it happened, they had a 20-foot container on board with, refrigerated aquariums, and they stuck the whole sample in there, and it spawned.
Wow. All right, so this is sample 1 for the expedition, yes? All right, so this is sample NA167-001, paragorgia with zoanthids going in the starboard, one of the starboard bio boxes.
Good job, Ayla, logging your first sample.
That was a horrible job. That was not a snip. That was more like a grab.
Hi, just confirming which box of the starboard bio box we put it in. What letter?
I don't know. You make a letter up. I think I would see. I can so the story is the sample box gets taken apart and put back together. So and it's always in a different order. So at the moment, it looks like A is the starboard aft. Or sorry, inboard aft, the little box aft. Looks like B is the second one. Looks like I just put a sample in C. So that would make the forward little box D. And then E would be the back right one. There should be a, May should have like a decoder ring up there for it.
Yeah.
It's the second one aft though, right?
Yeah, second one back.
Second aft.
Yeah,
Yeah, you can go ahead and close.
if you were to look on the sat feed 3, I have that image up to see the letters a little better.
So I'm going to leave this manipulator live. So if you touch that button, it will bite you. And I'm going to go off comms for a second and step behind the console. There's maybe I can get Mike to do it. Data lab, is Mike around? Or what do you have to call the data lab? Can you get Mike to remote into the NDI decoder for the cinema camera and reboot the thing?
You want me to head down?
You should be able to call him on the comms.
Oh, you don't have him over
lab.
there. Okay.
Oh, I can try as well. He might be at the video desk, and I'll try that.
Can you do it?
Yeah.
Let me try.
Yeah.
Amber has all the things.
I could crawl back there and pull its cable out and reboot it, but that's pretty brutal. Kind of like my sample. Give that box another bump in. Just oh, it's all the way close. Sorry. So you can swap the camera back now.
All right, I think we're ready to proceed upslope.
Roger.
Yeah, I think he's in the fishbowl.
Looks like just above the lasers here, another base of an old sponge.
Yeah, I'm gonna see.
You can tell here in the sand here, you see the ripples, which is an indication of current flow. And so even down here at, what are we at, 1,531 meters, there's still some current flow here. And you know, geologists and sedimentologists way more experienced than I am can read these things and get an idea of direction and stability of flow and stuff like that. You can pull a whole lot of information out of the bed forms here. But just looking at it, it looks like it's somewhat variable, and that the ripples come from different angles and they seem to be flowing around the different rocks.
So one of the scientists from Hawaii was saying that there's actually wave action down here, and whether the ripples are symmetrical or asymmetrical to determine if it's current or waves.
Yep, absolutely.
I've
That is
never
true.
been able to work it out, but
Down on the bottom right, I think I saw one of these crabs that carries an enemy on its back. I don't know if we could pan down a bit to see that.
Backpack crab, cool.
But yeah, the shape of the ripple itself shows if the flow is unidirectional or bidirectional. So if it's kind of more symmetrical, it means the wave is flowing back and forth across it. If it slopes up one and it has a steeper drop off on the lee side, that's unidirectional flow. Generally, if it comes from a current. Looks like we've got a decorator crab here potentially.
I am touching somewhere here. Let me, why can't I get down there?
I'm here in video. If you need anything, let me know.
Herc's touching behind, so I'm looking downhill, so I
Quick
can't.
zoom's fine.
Yeah, go ahead. If you're, that's good. I've kind of stirred it up a little because the back of the ROV is touching.
But here you can clearly see on the back of this crab is a sea anemone, and they actually carry these with their two back legs that have been modified. And the hypothesis is that it's sort of like a defense mechanism. They use the anemone's stinging tentacles sort of as a shield for themselves. And it allows the anemone, which would normally be stationary, to sort of move around and potentially have access to different food sources.
And this is a close relative of the hermit crab, correct?
Can you, while you're
I'm
sitting
not
there,
sure,
can
actually.
you check the gauges and do a
There's
recording
multiple,
for me?
I know hermit crabs do
I've,
something
glanced
similar
at
with
them, but I
anemones,
haven't,
but I don't remember
I
if
haven't
there's
tasked Jaden with
this is a convergently evolved group.
Right, are we
We
good with
we
this?
haven't written them down
Yep,
since
we're good.
upside down, and I haven't asked her to. It's just been, I didn't want to overwhelm her.
I always thought of those as anemone backpacks on crabs. Kind of cute.
You did? Oh, she did. Wow. I didn't even notice. Are they all good? Sweet.
All right, Dan, we're good here. We can continue on.
Roger. Okay, you can go wide, please.
Backpack crab.
We saw a couple of these on the National Geographic baited camera survey as well.
Do we know what they eat?
Well, they showed up to our bait. So many crabs in the deep sea are scavengers, except the ones that you see perched on top of the corals. Those are most likely using the corals to get up into the water column and feed on detritus or perhaps even associates of the coral. But the crabs that you see crawling along the seafloor, those are typically eating dead things that have sunken down from the surface.
I'm going to fly
So
up.
we landed on
Fly
kind
up
of
the
this
Nazarene to the
spur
north here.
coming off the side of the seamount, kind of a broader flat area. And we're just beginning to kind of get off this flatter patch and expect the steepness of the seamount here to increase as we move up towards waypoint 2. And all of this black rock you're seeing is manganese polymetallic crust that precipitates out of the water and coats anything hard down here.
There was some manganese encrusted sponges back there too.
Yep, those are old fossil sponges. So we got another sponge here, coral.
Always got to look to see if anyone's home. I don't see them.
So I believe this is some type of black coral. The yellow coral here is black, and it gets this name because of its skeleton, not its flesh color.
It's very Halloween, very creepy thing.
You wanna, hey Chris, can you home the DVL before you go? I don't know if doesn't seem right.
Oh, you're going to hear some new voices here in the control room for the next few minutes as we start shifting over and taking a break for lunch, or dinner, excuse me. So Harold is, whatever meal it is, whatever day it is, whatever time zone it is, I'm completely lost.
Let the watch cycle begin.
Where are all your fish, Jeff? There was. I know.
Oh, you're muted.
Was that the one you were looking for? Okay.
Yep.
Yep. Up.
Okay, wait. So which is the ultimate fish wish list?
So the fish on the wish list is this little rat tail. It has
Okay.
a long, skinny, tapering tail, and it has a really triangular snout, almost pointy at the very front. And when we were here in March deploying the Beta cameras, we saw tons of them, right at about the same depth that we're at right now. But we still can't figure out what it is. We know the genus, but we don't know the species. So I'm really hoping we find a little one. We can
Let's
get
look
some
at the
close-up
Atlanticella.
zooms.
You'll have to speak English, Megan.
The big white fluffy looking thing.
Thank you.
Tell me more.
This is a glass sponge in the family Euplectellidae called Atlanticella. I just like giving Dan a hard time by feeding him the Latin name first.
You know, I failed Latin horribly. And then made both of my children suffer by taking 2 years of it.
Anybody else take Latin in the room?
Yeah, if we do a quick zoom, you can see the lattice work of the way the spicules, the skeletal structures of the
go ahead.
sponge
Show your boots.
fit together.
Beautiful.
It's really cool, and these sometimes you can see they'll have associates living in them, polychaete worms, crustaceans and stuff, and you can actually see the animal running around inside the sponge through the kind of semi-transparent lattice of spicules.
Yeah, and we were also asked to collect snails that are associated with these sponges, but I don't see any. All right, I'm good with the zoom. Thank you.
All right, we got some follow-up questions on your Coelurynchus rat tail.
The Coelurynchus.
How many different types of Coelurynchus rat tails are there?
Oh, wow. There are, I think, something on the order of 45 species. It's a lot.
Okay.
And there are at
Dan,
least
do you wanna get
3
moving?
different kinds that we saw when we were here in March.
Okay.
But
good
this
for,
dark one that
we've
was
been doing
super
20-meter
common,
moves because I still got
we
the
had
training
never
wheels
seen
on.
before, so we weren't sure what it was.
Mm.
So, all the world over, rat tails in general, there's at least 250 species. It's one of the most diverse groups of fishes in the deep sea. And it's really interesting. They evolved specifically to be in the deep sea. You don't find any rat tails shallower than about 300 meters depth.
Do you guys have the NDI stream for the lower camera displayed back there somewhere?
No,
Okay.
we're using your screen. Can we take a look at the sea cucumber?
The Paleopatites.
This guy?
The one out there
Yep.
in the
There's the purple one. It's over off to the left.
purple one.
Yeah, that
Sometimes
other
I
one
wish I
was
had
a
a
Sericolophus.
Telestrator
It's
for
called
the...
something else now. Oh, there's a fish. Jeff, do you see?
I see.
Do you want to chase?
Very,
Let's chase
very
the
small
fish.
fish. Let's go take a look at the fish.
We'll be back for you, purple sea cucumber.
The sea cucumber will
Look at you
still
in a
be
moment.
here. Yeah.
Fish will probably still be there. I can see the fish, so I'll go chase the fish.
Chase the fish. We
Roger,
love fish.
fish herding.
The fish guy's here. We gotta chase the fish.
You see him in the upper camera there.
Oh,
So
yep.
he's up above the main camera. Let me come up and he'll magically come into view here. In theory.
There it is.
There he is. It looks like a little cusk eel.
Bazizitas? Seems
Uh,
kind of shallow.
Yeah, I think it's too shallow for that guy. We need a side shot.
You'll have to come down a bit for me, Sarah. To give
It
me
doesn't
some
look
leash.
like the pectorals are got the dual lobes.
Uh-uh.
I don't quite have the tether to come around there, but he's cooperating. Sorry, the fish is cooperating. I gotta break that habit. Or the eel is cooperating.
Yeah, I'm not sure which one this is.
Neither.
Is there any possibility of slurping that fish up?
Of what?
Catching
Capturing
it.
the fish.
Do you think you can get the net out and catch it?
No, maybe not the net, but what about the slurp gun? He's really small.
It is really small. What do you think, Dan?
Yeah, maybe. You'll have to move me closer though.
We are moving.
Roger. I'm at the end of my leash.
Just kind of waiting on Atalanta.
Roger. Well, while we're doing that, we'll see what we got for suction jars. See if I can remember how to turn that on. So I'm gonna get my co-pilot to do it. Where is that thing?
Brian, what is over on the side, the tumbleweed-looking thing, just to on the left side of the frame?
Oh, the sponges. They're called the catcher mitt sponges. They were called Sericolophus, but I think their name has recently been changed to something else. taxonomy is always changing. It's challenging to keep up sometimes. But we've seen these those sponges in Hawaii. Sometimes they grow in these pretty dense beds where there's a whole bunch of them. They root in the sediment, so they have these really long spicules that'll go down into the sediment to keep the sponge in place.
So I've, you still see the fish around there?
Yep, it's still there.
Still there.
So we've run some suction through the, what we call our flush jar there, the one with the red tape. So Jaden, now we're gonna press a button here, if we can find it. Sample jar, and it likes to rotate one way better than the other. I forget which way that is. We'll rotate around to sample jar number 1. Then if we somehow miraculously get this fish in the jar, you'll hit sample jar rotate counterclockwise, and that'll rotate the jar back so it can't swim back out. Still see the fish?
Yep.
I do not.
It's right up there.
Right. Still probably don't have enough leash to go fish hunting.
Yeah. Come on, Atalanta. You can go.
Fish herding. Well, I get to try my new 3D printed suction sampler, grab handle, and fish tickler. So Megan provided the design here for the TPU printed end effector on our suction sampler. Do you see where it says porch out there somewhere? Can you give that a bump? And one more. Okay. Oh, that was very brutal. Give her another bump out for me. Okay, now you can give it a bump in, see what happens.
I think I'm going to move you another 10 meters.
Porch in.
Bridge nav.
Yep, keep coming, keep coming.
Bridge nav. Yeah, can we move 10 meters north, please? Thank you.
Is that porch all the way in now? Right. So I'm just going to turn the, I'll be ready to, so you see the suction pump button here? So as I click it, it's on right now, but the longer I hold it, when that blue bar goes, and when I let go, it's on all the time. But Hercules doesn't have 1,000,000 horsepower, so that's taken, well, sorry, let me Hercules has 1,000,000 horsepower, that's taken 999,000 of it to run that pump. So when we get close, I'll ask you to turn on the suction. And it's really, I got it turned all the way up now, so it's really ripping.
Megan, do you still see the fish? We lost it back here.
Yeah, it's in the upper camera, you see?
Well, that's a snapper branched.
It's also in the
oh, it's not the same one.
That's not the same fish.
Oh.
What?
No, that one looks way bigger.
Yeah. I thought I had it, and then I realized I was just staring at a rock that wasn't moving.
Oh. Yeah, well, maybe we I think it went off to the
Would have gone
upper
off.
left.
Yeah, to the
Agreed.
left.
That's where it was last I saw it.
Probably heard me turn the suction sampler on and said, I'm out of here.
Yeah.
Well, and I would vote we keep our eye out for it and make some distance.
Yeah, I agree. Oh, there it is. There it is.
Or not. Push and drag.
We're actually still waiting for the ship because
Okay.
I am currently 25 meters away with a 35 meter tether.
Then yeah, then it's just off screen left in that sand patch.
Down in the hole.
Just to make your life that much more interesting.
Much more fun.
We want to give Dan some fun.
This is all part of the strategy where it's kind of a rock triangle there. We're herding them into the
Into the good zone?
into the cattle chute, sample zone.
How many different kinds of cusk eels are there? There's 250 rat tails.
It's in the deep sea, I think it's less, like we might only have 150 described species, but
Okay.
the problem is there aren't very many people working on this group,
Okay.
and we're realizing that they don't have a lot of external features that make them really easy to identify, and there's probably new species all over the place.
Okay.
So this is one of the reasons why it would be wonderful to be able to sample this fish. There's a pretty good chance it's new to science.
Awesome, thank you. I'm going to hand you over to the very capable hands of your SCF. Let's go, Elle.
Okay, you can go ahead and, well, sorry. Let me just put this down in a zone where I can smash it against the rocks. Can you come down on Atlanta, up 5 meters? Yeah. Sorry, you're gonna have to move us closer, Megan. I'm fighting the tether here, so there's no way I can. I'm now facing Atlanta. And, double tap your auto heading. I'll do it for you.
Can we move 20 meters 330?
So you're gonna stay on that heading, facing away from me for now, because I'm facing you. When you face away, that gives me the length of Atlanta, more tether, which is just enough. You saw how the ROV was getting all spastic there. It's because they were pulling on the tether really hard. I have to, I need this heading to look uphill. Obviously, I can't look downhill and be like reaching, down the stairs. Yeah, so in general, we try and stay in double digits with Atlanta altitude. So you're right there. You're 10 meters up. If we had any weather, it would be, it would look really scary. Okay, you can turn the suction on. Well, I don't know why Herc is porpoising so bad here. Roger. Yeah, they can feel when I get close and they run. Kind of need a funnel on here to do this. I have done it before with a funnel. Oh, there's your first TPU versus rock, and it's still on there.
Right, that's a win.
This is a really good example as we're looking here about mono versus stereo vision and how.
Oh,
Got it.
we got him.
Is he in the
Sample.
jar yet?
Oh, he's
There.
in there.
In the jar.
Yep.
Very nice.
Go ahead.
Oh, that was fantastic.
That was really
Very
great, Dan.
impressive.
Thank you.
That was just pure luck
And persistence.
We'll take it.
and patience on your part. Thank you for being very patient.
What's the sample number?
Yes, please.
That would be sample 2,
Sample 2.
Yes, sample number 2.
cusk eel, and jar 1.
What's that? Yep. Yeah, you could bump it
Is
again
it,
that way if you wanted.
This one?
We did it. I'm so excited.
Is
Jeff's
porch
day has been
all the
made.
way in still? Must be. Roger.
And there's cake.
Ooh, that's very hot now.
Oh, okay.
So I gotta turn that back down to
Yeah, I mean, you go eat.
less ludicrous speed.
Whose birthday is it?
I don't know.
Yeah.
And if you want, you can change the jar camera back to the rail camera, please. The current seems to be pushing us that way.
Yeah.
Yeah, so what we'll do now is we'll get it back in the box, as I say. So I'm going to come back towards you, and you should be able to bring your heading back around to the north. And then also, I'm going to come back down a bit, but you can also come up. I've come up a bit just to give myself some leash so I can. See if I have enough to. So if Herc gets tail to tail and like pulling on Atalanta, we actually get stuck. We get, if the ship's moving, we can't, get back around. Highly embarrassing because just get drug around all over the place.
So we have a question here. If the scientists could again explain why are we taking samples?
Harold, do you want to take this one?
Yeah, that's a great question. So we're in a completely unexplored part of the world right now. Here in Palau, there's been some research in the very deepest parts of Palau's waters in the trench, as well as a lot of work in the surface waters.
Is that a ctenophore?
And when we go to a new place with this technology, and we see things that have never been sampled here before, we need to collect what are called voucher specimens. And that basically allows us to get an identification on some of the things we see on the video. Because sometimes video is not good enough to allow us to identify something to species level. And if it's a new species, the only way to know for sure is to have a physical specimen. So going to a new place like this and collecting specimens is absolutely essential for discovering new species and helping us identify all of the things we see on video. And sometimes species can be cryptic as well. You can't tell them apart, even if they look completely identical on the outside. You need those specimens to be able to do genetics and morphology and we only need
You could probably come
one
up
or
with
two.
it there if you want.
So it's a non-destructive activity.
And Harold brings up a really important
Somewhere
point,
in the
thinking
low
about a
teens
lot
for
of
altitude.
less invasive technologies like environmental DNA and things like that. You have to have the reference libraries for that. And so, and you have to have described the species. So we still
you hear
need
me okay?
physical samples in order to
I can't hear
identify it. Then if it is new to science, be able to do a really detailed
you gotta
description
put
of
that mic,
what it
like,
is so
real
other people
close
can
to your mouth.
identify it. And then
You got to speak up too.
use those to build
Yeah.
a reference database for use
Yeah.
of genetics. So we can come down and collect water, look for the DNA that's floating around in the
You
water,
could
but we
come up
can't
5
use
meters.
that to identify what species are there unless we've collected the species initially, done the genetics on it, and have that in some kind of reference
What's that?
library.
Come up about 5 meters. Try and keep
Looks
that
like we've
delta
got
in
a
the,
rat tail here with,
yeah,
looks like a parasite on the side of it.
in the teens, the altitude in the teens.
As well as another paragorgia covered in zoanthids.
You let me know when you're ready to start moving again, Dan.
Ready, ready.
Roger, roger. Bridge nav, 20 meters, 020.
Can we zoom fish right before we look at coral?
Sure.
It's just behind the rock with the little lollipop sponge on it.
Where'd it go?
You got it? It's right.
Yeah.
Go
It's
ahead.
right in there.
Okay.
So you can see on channel 3 when we want to focus on something, the scientist will have a blue circle for zooming into things that are on camera.
Is that rat tail feeding?
Is that a lollipop coral, you said?
It's a sponge,
A
and
sponge,
that's
lollipop.
not really a technical term. It was just a quick description of it.
I don't.
It's another type of glass sponge. You can also see it's got two isopods living on it.
I don't think I've ever seen a rat tail actually eat anything.
Not sure it's
No
eating.
wonder they're so skinny. No,
A rat tail is a fish?
Yeah, the rat tail or macarorid
Okay.
is the fish hiding behind the coral there, and then it's got a parasitic isopod living on its flank.
what is that behavior?
I
I really
not entirely
don't know.
sure.
Looks like it's just sort of swimming into the ground.
I would say it might be avoidance, but
Hiding.
all right, you got what you need here? Okay. All right, can we go back and take a quick look at the coral and the other sponge on the rock to the left?
Roger. I thought you were going to wow me with some scientific terminology there, Harold. Swimming
Every time I
into the
try and
ground.
use fancy terminology, someone tells me I'm wrong.
Yeah, these rat tails are something we saw very often in our baited camera survey. And there's multiple different species, multiple different genera that we're expecting to see on this mission. And having that close-up zoom is very helpful to go back and confirm some of our identifications from our baited camera videos, because those cameras stay in one place and you can't zoom in on them like you can here, so that's very helpful.
Might get a still picture there in our very laggy still camera. You can zoom right in there if you want.
For video?
Yeah.
Okay. Did Mike come up? I sent him your way.
Did that camera just turn itself off? Or is-
I just toggled into autofocus and it blacked out for a while.
really?
I'll let you play with that. Yeah, looks very similar to the paragorgia we just collected with the same kind of gold zoanthid, parasitic zoanthids overgrowing it. I don't see anybody living in this one right now. Can we pan over to the sponge to the left, please?
Roger.
Right. That's probably good on the main HD. Harold, you get us a couple pretty shots on the still cam?
I think so.
All right, let's move on up then.
Roger.
Maybe we'll make it to waypoint 2 by the end of our watch.
I'll try and center that thing up as I'm coming out here. No promises, but I want Herc.
So just to explain to some of our new viewers, we are
Take a few more stills there if you want us to walk around it here on the
we
rock.
are 45 nautical miles north of Velasco Reef, which is part of Cayenne up north, and we are exploring an unnamed seamount, and
Right, Dan, we got a couple shots.
Roger. It's that one. Need to move that camera forward a little. I see what's happening. There's certain, sorry, I'm trying to get it back to Jonathan prefers a certain zoom level, which happens to zoom it in enough not to see the bio box in the porch.
Oh, you want me to zoom in? Sorry.
No, not you. I was playing with the lower camera there. He likes it on whatever, it's 7 millimeters, some shutterbug number that equates to some number here. No, he likes it on 11 millimeters, which equates to -7 on the zoom. I think, I don't know. He'll chime in if he's listening.
It's interesting. I'm seeing a lot of these dead sponges on the ground. They look like they might be Farreidae, perhaps, but I don't think I've seen any
Good
live
20,
Farreid
Chris.
sponges.
Yeah.
So maybe
Bridge nav
long ago there was a
2020.
sponge recruitment event where they were thriving under whatever food or current regime was active in this area, and then maybe conditions changed and they died off. It's hard to say.
And that's interesting, something we see not infrequently in the deep sea, where we see a whole bunch of dead skeletons of a type that aren't present now. And with the long lifespans of these creatures, you know, climactic events, paleoceanography can really control these distributions. And when you get large-scale recruitment events and have really favorable conditions that may not exist in today's ocean, sea lily coming up here, or stalked crinoid. I always find these to be really nifty creatures. They're quite prevalent in the fossil record, going back hundreds of millions of years, and it's only relatively recently that we realized, that we started exploring the deep sea, that they were still alive or extant. They thought to be literally a fossil relic of a bygone era, and as we started to explore the deep sea more and more, we realized that a lot of the species and groups that we only knew from the fossil record are still alive and well in the deep sea.
Megan identified this one earlier, and I'm trying to remember the name of it. Something rubrum. Something crinus rubrum.
I was trying to get the front of it there in the still camera, and I blew it away. I was going to come back around for you there.
I'm going to try and get a quick picture here.
Yeah. Got like 10 of them. Maybe one will turn
I think
out.
this is some type of parasicrinus.
You keep bashing that button, I'll keep moving closer. And the camera freezes. We definitely got some work to do on that.
I think we're good.
Right.
Good to keep moving, Dan?
Yep.
Bridge nav, another 20 meters.
Looks like another metallogorgia, another big grass sponge.
I think Jake was right. The vehicle definitely feels a bit on the heavy side.
This is a euplectellid sponge, similar to some we've seen already.
Another note in there, Jaden, is the lights, the mids need to be adjusted in. It's really dark with the down lights off.
We have some new viewers watching, and they're curious about where each of us are from and
Dark spot
your
there
roles
in the upper
in this
camera.
expedition. So could I have the back row start with the names?
Good for another
Sure, yeah.
20, Chris.
My name is Harold Carlson.
All right.
I am in the science chair for this watch from 4 to 8.
Another 20.
I'm a PhD student at the University of Hawaii at Manoa, and I study deep sea ecology, specifically deep sea habitats like this one and how fishes use them.
And I am Jeff Drazen. I am a professor at the University of Hawaii and also a deep sea ecologist, and I specialize in understanding deep sea food webs and particularly the ecology of deep sea fishes.
Ali, and hello everyone. I'm Ayla. I am the data logger on this dive, and I'm currently a Marine Lakes Research Assistant over at the Coral Reef Research Foundation in Palau.
Thank you. Ali, everyone. Hello. My name is Elle, and I am the Science Communications Fellow during this voyage. First, to start off, we'd like to welcome all the new viewers that are coming in, and we are reading your questions, and please chat in the box if you have any other questions about the dive. We have, in our front row, we have a navigator, 2 pilots, and our videographer.
Yeah, they come up. Up 5.
We've got an interesting sponge coming up here. You can see that enormous base of attachment there. All of those fibers coming down. I believe that's called lophophytus attachment, if I remember correctly. But wow, yeah, you can see how it's just like hairs coming out of the bottom of it. And it's just sort of this half dome.
Push in a bit there.
What did
I'm
you think
hoping
it was again?
Video.
So this is a glass sponge. Trying to look into a better ID. Hoping Chris Kelly is still online. I haven't seen him in the chat though.
This is the ROV pilot feeding a glass sponge.
I'm going to reset
This
your
looks
TVL,
like
okay?
it could be some
What's
kind
that?
of
You get your TVL
polyopogon.
reset?
Right. I'm going to try and zoom out on that lower camera a little and get the whole thing in the...
So those sort of tendril or hair-like structures that you're seeing now are called spicules. And in glass sponges, these are sort of like little spiny structures that make up their skeleton, and they can be partitioned into different parts depending on where they are in the sponge's body. And I've never seen a sponge base quite like this, where it's just sort of this lion's mane of hair coming off of it, attaching it to the seafloor. It's pretty cool.
Okay, can go wide, please.
Could those attachments mean that it's holding on so it doesn't, if there's a current, or what could that mean?
Yeah, that's exactly right. So sponges are what we call sessile, which is just a fancy word for they don't move. That's because they're filter feeders and they like to hang out in the same spot where they're going to get a relatively consistent supply of food coming from the current, delivering particles, zooplankton,
Let me get
detritus.
up ahead there.
Sure thing.
Par right now. Get up to about here.
Okay. That DVL's all over the place.
This looks new here, this coral. If we could get a quick zoom on that.
Sure.
Oh.
Looks like a black coral, maybe a bathypathes.
Yeah.
Can push in a bit there, video.
Sure thing.
It's good, thanks. Happy with that? You want to?
Yep, that's great.
Okay, could go wide.
So the corals we've been seeing, are they soft corals?
Yeah, so soft corals, as I understand it, at least in shallow water, refers to octocorals that you would find on a coral reef. So these are corals that don't have a hard skeleton like the reef-building or scleractinian corals that we see in the shallow waters of Palau. But these corals that we're seeing now, many of them are related to those same soft corals, and we call them octocorals because their polyps have 8 tentacles, whereas the shallow scleractinian reef-building corals that you're used to seeing snorkeling here or in many other parts of the world, those polyps have 6 tentacles, so we call them hexacorals. And the black coral that we just saw, those are actually a type of hexacoral. So they're more closely related to shallow water corals than they are to some of these other deep sea corals we've been seeing, like the bamboos and the chrysogorgids. So the amount of coral diversity down here is quite astounding.
You snapping stills there?
Sure. We've got some kind of sponge.
You good? Moving on.
Yep, we're good.
All set.
Could I ask if somebody could explain the 2 green lasers that are on the camera?
Sure. When you only have one video window, it's really hard to tell the size of objects. So basically those 2 lasers are 10 centimeters, or about 4 inches apart, and we're shining them right on a shrimp right now, which looks like it's probably called nematocarcinus. We probably want to zoom on that guy really quickly. Oh, there he goes. But it's just a ruler that we can move around anywhere where we're looking, so we can get a good idea of the size of things.
Can do the lasers in
yep,
there.
That is definitely, it has really long, spidery-like legs. That is the genus nematocarcinus.
Sorry,
and we see these are really fantastic shrimps. We see them in quite a few different habitats in the deep sea.
It has some long legs.
Really long legs. They're usually 2 times the length of the whole body.
Olek. So Olek is wow in Palauan, so.
Okay,
yeah.
Great.
These are another animal we saw on our baited camera deployments showing up to bait as scavengers.
Got a lot of legs.
That's a good zoom. Thank you.
Okay. Off we go.
We have an ID in the chat from that black coral that we saw earlier from Tina Molitsova, Bathypathes pseudo-alternata. Pseudo-alternata, excuse me. Thank you, Tina. As we look at this bamboo whip here, we're seeing some interesting geological structures in the background, these fissures running along the seafloor.
Got a shrimp there hanging onto the side. Another little cup coral. A couple different little things hiding around on this little mini promontory.
It's interesting that of the corals that we've seen so far, the cup corals are the thing that's most closely related to the reef-building corals that we see on the main islands of Palau. And down here, there's quite a different suite of corals.
I don't actually know what depth the aragonite saturation depth is here, but I'm sure at this depth we're below it. So it's really hard for the sclerotinians to get a good look. Can we take a look at this one? I don't know if we've gotten a good look at this variety yet.
All right.
This this sparsely polyped Chrysogorgia. Caught a glimpse of it earlier, but I don't think we've gotten a good look at it yet.
Those are so beautiful. I love those ones.
Okay, Amber.
They're really easy to miss too.
Yeah, I can't even see it in my monitor.
Yeah, they make
Oh my
doing
gosh,
video
it's
annotations
invisible.
very
So
fun.
delicate.
Now let's see if it's got a shrimp. These corals frequently have a shrimp associate hanging out in them, probably near the top. Pretty good current too going on down here. Coral is pushed all the way over. You can see all its polyps out. A lot of the octocorals, interestingly, feed downstream, and that their polyps are located or tend to be streamed away from the current with their feeding tentacles facing away. I've always assumed it is taking advantage of some of the hydrodynamics of it, using its own body to slow down the current flow, causing some turbulent flow that might have some little eddies or something, that then the tentacles have a higher likelihood of actually getting a hold of the food particles. Can we pan a little bit left and see the top of it? I want to see if there's anybody home.
I feel like I have to describe that in sports metaphor. It's like it's taken the physical charge so that it can get the feeding.
One of the little benthic
Ctenophores.
It looks like it might be an amphipod. I see two amphipods.
Where?
We've got one, can't point to where they're going. Right there and right there.
Yeah, you're right. Those are amphipods.
Yeah.
Yep. All right.
I don't.
How teeny tiny are those?
They're certainly well below a centimeter.
Yep.
You know,
Quite
I
small.
measure them in millimeters. All right. I think science is happy. Thank you, Dan.
Roger.
How about in grains of sand?
Surprised we hadn't seen any of those long-clawed shrimp that are real common in those.
Good for 20.
Roger. Bridge nav.
Yeah, we
20
did see one of
meters,
those
020.
in one of the earlier Irritagorgias towards the very beginning.
What's the Latin for those? I don't remember.
The shrimps, I don't know.
Yeah, I don't ever remember Latin.
They're pandalids, right? Is that the family?
I truly do not remember. I have to go look. That doesn't sound right to me, but.
Okay.
So for all of our viewers at home, how do you even begin to learn all of these? Is like learning all the names something you've always been into, kind of categorizing things, or what's this process like, knowing that you all are truly exceptional at it?
Well, it's not easy. And it's really just a case of practice makes perfect, like it is with many things. So I sort of got involved in doing this kind of deep sea research through the NOAA Hollings internship. And through that program, I worked with somebody doing a project with the NOAA Okeanos Explorer, which does very similar style of exploration with ROVs that we're doing right now, and worked on a video annotation project there and used some of the resources that they have online, like the NOAA Deepwater Benthic Animal Guide, to study ROV images like these.
Can we collect this?
Yeah.
I'm pretty sure it's a
bridge, hold
bathypathes,
position.
but the associate brittle star is unusual.
But yeah, it's
I
mainly just
deploy?
looking at images like these and developing a well-trained eye.
As someone who still really struggles with the Latin, I'm open for suggestions because I'm terrible at it. By the end of the cruise, I'll remember it and be back to being able to do the Latin for most of it. But then 6 months from now, when I get on the next boat, I'll have to start over. My brain just does not hold the Latin names well at all.
Well, if it makes you feel any better, I took Latin in high school and college, and I don't think it's helped me at all learning these names. So,
Do you want the entire
I think
animal
the
or-
thing that
Yeah, because this one's going to be particularly hard to cut and probably be floaty. So I think you're going to end up probably having to pluck the whole thing off the seafloor.
Right.
But I think the thing that helps the most with learning all the names is to create a guide. It's
you're
just
fine
like
there.
if you were to pick up a bird guide or something like that on land, and by flipping it through it and getting an idea of what all the animals look like, you start to organize those things in your head. And so usually after these expeditions, when we go through and we actually process all of the video and convert it all into counts of different kinds of animals, we create these animal guides. And so it's kind of a little hard going into an expedition because you don't know necessarily what you're going to see. But certainly by the time we're at the end, we've got this. It's basically like a bird guide. It's just a deep sea animal guide with all the different kinds of animals. And making those really helps stick all the names in your head.
And one of the things that you'll, if you really get into it, you'll notice we change what level of taxonomy we are working with quite frequently. Sometimes we're talking in the class level, sometimes we're talking at the family level, sometimes we're talking at the genus level. And so that's another thing that makes it a little more complicated. You'll hear some of us, if something we can identify to the genus level, like the Irritagorgids, you'll hear us say Irritagorgia, even though that is, a genus under the family Chrysogorgiidae. And so, and a lot of the genus and family names are quite similar, like just to make life that much harder. And then it changes. And there was a major redescription or reorganization of the octocoral genuses and families a few years ago that's still slightly up for debate. And so a lot of us who learned one set of taxonomy are having to adjust to a new set of taxonomy relatively recently.
So what are some tips and tricks to remembering scientific names for different species?
well, I find that the ones I remember the most easily are ones that are sort of memorable or silly or ridiculous. Like trying to think of a good example. There's a genera of zoanthid, the parasitic polyps that we saw earlier, called bullagummy zoanthus. That's been a new favorite of mine because they grow on bubblegum corals, so which is already a funny name. So, I mean, it's not everything's gonna have a fun, silly name, but sometimes the taxonomists they insert a bit of humor. And,
Yeah, that's pretty cool.
yeah.
Zoom in there. Full zoom. Floaty. Where do you want? Try and put the floaty.
So that's a little.
It's your. I don't have a strong preference. I think this is going to be a bit hard because they are floaty and sticky, and with you've got a fair amount of current. So wherever you think is probably the safest place to put it.
I think that's partly the ROV. I'm just gonna put my nose into it here. We might play around and disable the birds here.
So this is a black coral, likely a bathypathes.
And we have confirmation from Tina and Asako in the scientific chat that yes, it is bathypathes. Cool. And Tina would like one pinule preserved in formalyn.
Right.
If that's
Can you make a note on the sample sheet, please? And this is a ophiuroid or brittle star there, right in the right next to the claw of the manipulator arm. That's not one I immediately recognize, and it's a general form I'm not used to seeing growing on corals. It's not the kind of normal, usual suspects for coral associates, which is the primary reason I wanted to collect this particular one, is because the association is not one I'm used to seeing.
Come on, Herc, find a spot. Park it. Park it, Herc, park it.
So what we're doing right now is we're just changing the position of the vehicle to make it easier to get the sample into the bioboxes. Black corals are notoriously neutrally buoyant, and maybe even a touch positively buoyant. And the mucus that comes off their tissues when they're distressed tends to be pretty sticky. So trying to get these into the container can be a challenge, and given that we've got a moderate amount of current flowing by, this is going to be a little bit on the harder side. So we're trying to get the vehicle in a position that'll optimize our likelihood of getting it successfully into the box.
It would be, I can put it in the front box pretty easy.
Okay.
Then you have, I don't know if we have the fish tanks in there or not. One way to find out. So the forward box, I'm just going to bump it here. See what's in there. Looks like fish tanks in there. Then typically when we're playing around in the front there, we retract the HD camera like so. You can go wide now. That makes it less likely for me to poke my eye out. Oh look, there's a scoop in there. And for floaties, it's easier to have a floaty in the front box because we can see really well if when we open it again, whether it's going to float out or not. And we can quickly close it.
All right.
Why is that scoop in there?
This is going to be sample 3, correct?
Yes, sample 3. Forward bio box.
With a
So what I'm going to have you do, Jaden, is just bump that box closed. Just kind of bump, bump, bump till it is trying to pinch the manipulator. Yeah, you can bump it again. Keep going until you see it. Kind of hit there. Yeah, so then it do a bump out. One, yeah, maybe one more. Okay. Well, that lets me kind of tuck the manipulator underneath here and let go and hope for the best. You have to do a bump out now for me. One more. See if I can get out of there elegantly. Nope.
Excellent.
That doesn't appear to be floaty. Well, maybe.
Yeah, it's not.
Yeah, go
Yeah,
ahead,
there it
close
goes.
the box.
Close. That's what you get for trying to prove me wrong.
Okay. I take that back. It does appear to be floaty, or at least neutrally buoyant. Very nice.
All right, sample
response on
3
closing
and
the box.
port forward biobox. Bathypathes with unknown orpheroid.
And it may be worth making a note.
Yep.
To hand over to the other.
That's floaty.
Well, they know because they rigged it. They have a scoop in the box, which is going to challenge somebody at some point.
Is that a predatory tunicate?
Where?
Top left. I'll highlight it.
Oh,
Can we go look at that, please?
Yeah. Is it anchored?
Looks like it.
Oh.
Yeah, I think we've seen a couple so far, but we haven't gotten a good zoom on one.
They're really.
I'm going to scooch us forward a little bit, Dan. 10 meters or so.
Roger. I'm coming towards that.
Bridge,
challenging
nav,
to
10 meters due north.
This is a good heading because we're in the current. Many times I've tried to get up close to them to get a good shot and fail because they're so delicate, easily blown away. Just gonna get the camera ready.
Dan, do you have a favorite environment to pilot in?
Yeah, but not a single one. I don't know. And generally, I like vertical. Cliffs.
I've got a large crinoid on the left as well. I don't think we've seen one quite that size.
I don't think that's a crinoid. I think that's a bracingid.
Bracingid. Oh, you think so?
Yep.
No, this is.
Well,
I was thinking this was what do you call
never
it,
seen a purple one.
Star or sea star. That's not what I was thinking.
Well, we can zoom on that next.
Yep.
What's the one with all the spider webs that anchors itself in?
Not sure.
The crinoids, the little bundle of legs?
Oh,
Are you in a brittle star? Basket star?
They have all the little spider webs. They're usually like in a little cave. They're really hard to see. You can go ahead and zoom in there.
Basket star.
I can't remember the name of them now.
Okay, what are the two camps? What do we think it is?
I believe it's some type of predatory tunicate.
Oh, this is the one they were calling the smiley football on the last expedition. It has an opening for water flow.
Yep. And it's got a filtration system basically inside, and so.
So this is an animal?
It is, yep. And amusingly, short of the fish, it's probably our closest relative we've seen so far tonight. It's a tunicate, so it's a chordate, just like humans are.
It's not a bad life. I like it.
I'm used to them having a clear clearer skin. Like this yellowish color is not what I'm used to seeing. Looks like it's got a couple things probably living on it too. You can. It's got two holes, one to let the current flow in, and then a set of filters you can kind of see in there, and then an X-current siphon where the water leaves.
So the two holes allow it for it to collect nutrients?
Yep. So it's just, it's a filter feeder, primarily.
Did we have an ID for it?
It's some type of tunicate.
Okay.
All right, science is happy with this one.
All right.
We take a look at the big purple, either percentid or crinoid to the left.
I'll scoot over there a little.
How you doing over there, Ayla? First data logging.
So far, so good. Still learning a lot, too.
Oh, constantly.
Okay, I'm gonna move the camera. Movement.
Did I jump it? Were you ready for me?
Totally looks like I was wrong. That's
I'm calling that a crinoid.
Nope, I was wrong.
Let's see if I can find a even closer ID for it. That's a good zoom, thank you.
So Daniel, is that, sorry, too many Daniels around. Dan, I guess you might also be Daniel. Is that what you were describing with its little feet? Where it has the bundle of hook feet? Or were you thinking of the arms that have arms that have arms that have arms?
Isn't it a tunicate that, we call it a sun star or a sea star, so it like makes this web. And they're really, you got to get really close to see all the, it's not a stopped.
Like a dandelion?
Yeah, dandelion.
Dandelion, siphonophore.
Yeah, that's it. Sorry, benthic siphonophore.
Yeah.
Yes.
That's a good zoom.
Moment there.
Thank you.
That's what I thought he was at first. Thought it was at first, sorry.
All right, I think we're good here.
Roger. We got a really fantastic shot of one last year, and that was the first decent shot. Like the 3 before that, you get close, and the thrusters just
Yeah, they're very
blow
delicate.
away.
In the Eastern Pacific, they have it's very different. It's also a siphonophore, but it's a different type of benthic siphonophore that's really freaky looking. Like it's even weirder looking than the ones we get on this side of the ocean. Big long bamboo coral whip here as well.
How old do you think that whip is?
You know, I mean, hundreds if not thousands, but I am very reticent to give you much more than that range because we really just don't know.
I mean, that's so amazing, right? We get sometimes used to that in the deep sea, but you're like, hello, 1,000-year-old resident of this beautiful place. Thanks for letting us visit.
Isn't that amazing?
Absolutely. Here long before the establishment of the Palau National Marine Sanctuary, hopefully here long